All right. So, the next section of the book starts to talk about how the brain changes over time and also kind of the biological basis for learning and memory. One point that it makes that is very, very different from what you would have learned if you would have taken this class 20 years ago is that the brain is constantly changing, developing, and growing over time. So first of all, for most of you watching this class, who are the typical age for freshmen, sophomores, juniors, or seniors, your brains are still growing. The prefrontal cortex doesn't fully kind of set in until about 25. But even for those of us who are a little bit past that, what we see is that it used to be thought that all of the neurons that you were going to have over the course of your life were created during development and when you were a baby, and that by the time you got to adulthood, everything's downhill. And what we see is that that's not the case at all, that you are still growing millions of cells and creating millions of connections every year. So the brain is not set in stone, it's still plastic even into older adulthood. Um, what we see is that we can catch evidence of the physical changes in the brain that correspond with learning and memory. So first of all, on kind of like a gross level, what we see is that if you use it, it will grow. In the same way that a muscle in your arm, as you use it, it will grow larger. The same thing that will happen in terms of neurons, except for a particular task, what we find is that actually it grows, but also it takes up more brain real estate. Um, Now, one of the easiest ways that we can see this in rats is in enriched versus deprived environments. So, you should always take a little bit of research with rats in question because the circumstances that we house rats are highly unnatural in compared to the rat's evolutionary history. So rats are designed to be social creatures in highly complex environments. Whereas for the convenience and cost, when we do research with rats, for the most part, they are in tiny little cages that are very boring by themselves. Now, this is one of those things where it creates less variability and removes variance from the equation. And so, from an experimental design point of view, it makes there less error and random things that are messing with your experiment. And it also just costs less to keep just a rat in a tiny cage as opposed to a giant rat utopia. But what we see is that it comes at the sacrifice of generalizability, that this is how rats behave in a very unnatural environment. If you put rats in an enriched environment, like for instance, when I used to work as a zookeeper, we had to do enrichment where we provided stimulation for the environments every day. We had to give toys to play with, or new foods, or new smells, or change things around in their cage. And the same thing with rats. If you provide a very complex environment, and particularly also if you give them a social environment, the brains of these enriched rats will look very different, particularly also their hippocampi will be much more complex and dense and even grow a little bit. And so you can see that the environmental effect of having more reasons for your neurons to fire being directly reflected in their anatomy and the size and density of the neurons and the connections in their brains. What we also see in humans, it would be unethical to just slap a human in a cage from birth. But what we do see in humans is that brain areas will be recruited the more that you use them. The sensory areas will be, and motor areas will be larger and more complex for individuals that are engaged in really fine motor tasks, such as concert violinists. Or what's really fun is to look at, for instance, the occipital lobe in blind individuals. So hopefully you remember from the previous video in the book that the occipital lobe is where all of our visual processing goes. I have no idea why they chose the very back of the brain to process the things that are coming from our eyes, but I didn't design the thing. Um, and what we see is it's not like that brain area just sits around going unused. That would be very wasteful. It actually ends up being taken over by different tasks that will light up in the fMRI machine for blind individuals that won't necessarily light up for sighted individuals. Um, you can also see this in hippocampal enlargement in chickadees and cab drivers. So chickadees or squirrels also bury seeds, and then they have to remember where they buried them. And what we see is that there's a direct connection between the hippocampus in chickadees and squirrels and their spatial ability in terms of burying seeds. Um, there was also structural differences that were observed in cab drivers in New York City. So this dates the research a little bit because before there was Siri and there was Google Maps, if you wanted to get around in New York City, like you either had to be really good at reading the map or you just had to memorize the whole bloody city. And it was a very complex spatial problem. And if you remember, the hippocampus is the seat of spatial reasoning and memory. And so they saw an enlargement in cab drivers in the hippocampus, reflecting the fact that presumably they had to remember a lot more spatial information than the rest of us. It'd be really interesting to go back and do the same research now because you don't have to remember where everything is. Now you just got Siri, and so to see whether that difference in brain anatomy would still persist, or maybe that's something that was lost due to the change in the environment. This brings us to instead of on the tissue level or area level, we can also see on the cellular level evidence of physiological changes corresponding with experience and learning and memory. So there's a phenomenon that we call long-term potentiation, which we believe is a big part of learning and memory. Um, as neurons fire in concert, the connections between them strengthen so that they're more likely to fire in concert in the future. Or, as one of my neuroanatomy professors would always say, neurons that fire together wire together. Basically, if I have two neurons that are firing in concert, where one neuron is firing and it's making the next neuron fire, there are physiological changes that happen at the synapse. So the presynaptic membrane, that little bud on the axon, ends up getting bigger. And it ends up having more vesicles and more neurotransmitters that are hanging around waiting to be released. And similarly, on the receiving neuron, you have an increase in receptor density. And so what that means is that, for example, if I go in and I electrically stimulate a group of neurons to get them to wire and to fire together, they will increase the receptivity there at the synapse, so that if I make one fire in the future with a smaller signal, it will still get the next neuron to fire. So you can literally see the wires connecting up in between neurons in terms of this long-term potentiation. We also have been able to manipulate this. So, for example, if I give you a drug that blocks the neurochemical precursors for this process, mice end up becoming dumb, where they're not able to benefit from experience and learn how to run mazes faster than if they don't have the drug. Similarly, we're getting really good in mice at being able to genetically manipulate them to express different proteins and different genes in different tissues. One of the things that was really famous in the late 1900s, early 2000s is Doogie mice. you are probably way too young to remember, but Neil Patrick Harris originally was a childhood star for his role in Doogie Howser, MD, where he was this like kid prodigy doctor. So they named these Doogie mice because they manipulated the genes for this long-term potentiation process. So the stronger version of this process would be expressed in the hippocampus. And lo and behold, what they found was that they produced Doogie mice that were smarter, or more specifically, were able to benefit from experience more and have higher learning than other mice. Now, one thing to keep in mind is that everything in the brain is in balance. And so, for example, in my wife's research, what she found is that there are this was groundbreaking research in the early 1990s and 2000s. Now it's just Tuesday, and the average graduate student is doing stuff like this, right? And she was studying a particular area of the brain called the hippocampus, and this particular microRNA called MIR132. And she designed, you don't need to know this necessarily for the test, but she installed a TET off and on switch where, using antibiotics, she was actually able to precisely control how much this gene was expressed in the hippocampus. And you get this bell curve where you reduce the natural amount of this particular gene in the hippocampus, and you get really dumb rats. And you turn it up just a little bit, and you get smarter and smarter rats. And then if you turn it up too much, you get really stupid rats again, because you have too many connections in the brain. And what we see is that this happens a lot in the brain, that there aren't necessarily absolutes. So we can increase long-term potentiation, and it will make rats smarter and maybe humans smarter, although you would have to be genetically manipulating children like at the cellular level from like the zygote stage. And we kind of decided that's not very ethical. But also know that a little bit is good, just like salt, but too much, and all of a sudden everything goes south. So that too much long-term potentiation is a really good way to have inefficient neural networks and seizures. So there's a little bit of a Goldilocks area there.